So welcome back from the break. Let's take a look at the two questions. First, compare the early development of a snail, a protostome, with that of a deuterostome, a human. So protostomes, in terms of cleavage pattern, are going to have a determinant and a spiral cleavage pattern. The human as a deuterostome is going to have a radial cleavage pattern and indeterminate cleavage. So remember, determinate means loss of potency with the immediate formation of the blastomeres. Indeterminate means retention of totipotency with the early formation of the blastomeres. The body cavity in a snail as a protostome is going to be schizocoelous. That means it's going to result from the splitting of the mesodome. The body cavity in a human as a deuterostome is going to be enterocoelous, a result of the pocketing from the developing archenteron in the mesodome that's going to give us the beginnings of the body cavity. Then if we think in terms of the fate of the blastopore, in a snail as a protostome, the blastopore is going to become the mouth and the second opening will become the anus. In a deuterostome, like a human, the blastopore will become the anus and the second opening becomes the mouth. So that's how you would do the comparison between a protostome and a deuterostome. How do animals that lack a body cavity, the so-called acoelomate body plan or compact body plan, exchange materials? Well, remember, this is going to be a very flat body plan because of the lack of cavity, which means that the body overall is only going to be a few cell layers thick, maybe 10 to 15 cell layers thick in some of the thicker regions. It's also going to have some form of gut cavity, as you'll see, that penetrates into this solid body plan or compact body plan. So there are going to be very short distances between the cells of the body and either the outside of the animal or the gut cavity itself. And so the animal can actually rely on diffusion, either through the epidermis, derived from the ectoderm, or through the gut cavity itself. Now, the lining of the gut cavity, which in this case would be a derivative of the endoderm. So diffusion works for something that is acoelomate, and we'll see that when we talk about the invertebrate clades that are identified as having an acoelomate body plan. More of that to come in the next chapter. So let's move on and talk about the diversity of the animal kingdom. So as I've mentioned previously, we do recognize the presence of about 35 or perhaps 36 phyla within the animal kingdom. The reason why I say 35 or perhaps 36 is because there have been some animals identified recently that do not appear to fit into any of the currently recognized phyla. And as a consequence of that, there is the possibility of creating a new phyla to accommodate these animals. There's a lot more work that needs to be done on this, but this is the nature of the beast as we are able to take a look into a greater number of diverse habitats, particularly in marine environments, where there's always a limited ability once we get below about 1,000 feet with the loss of light. It's very difficult to see past the limit of the lights that you have to carry with you. And that means you could have an animal swimming in the ocean no more than perhaps 100 feet from your observation point, but you'll never see them because it's dark. And so we are finding more and more that we are perhaps going to have to establish more phyla as we find or as we penetrate into some of these previously impenetrable habitats and start to identify more and more species and start to recognize that we're identifying species that don't really fit into the currently accepted phyla. That's something that has happened over the course of the last 10 to 15 years that I'm aware of, at least. But on that basis, we can go through and create a phylogenetic tree that looks something like this. Now, this shows the major clades. It doesn't show all of the clades, obviously. If there are 35, perhaps 36 currently recognized phyla, clearly they're not all present here on the phylogenetic tree. But this is the currently accepted phylogenetic tree of the animal kingdom, showing the major clades that we're going to discuss in the next two chapters. So what is this phylogenetic tree telling us? Well, here are some of the key points of that phylogenetic tree. And notice I've used the term hypothesis here. Remember, a phylogenetic tree is always a hypothesis. It's our best answer to the question, what is the phylogeny of the animal kingdom, based on the data that we have right now. So here are some key ideas. All animals that are known in the animal kingdom right now, all of those that have been described by science, form a single clade known as the metazoa and are derived from a single common ancestor. Sponges are still considered to be the basal animals and are a sister clade to the animals that contain tissue. So sponges are a sister clade. They are a sister clade to the eumetazoa. So eumetazoa means true animals. So that comes from the prefix EU, as a Latin term. The prefix EU means true. So when we put U in front of metazoa, we're talking about true animals. So true animals also form a clade. And these are the animals that have true tissues. So the name eumetazoa means true animals, but what it really means is the presence of true tissues. There are two clades that are radially symmetrical and are basal to the animal kingdom. So the radiata, Nidarians, Antennophorans, basal. To the bilateria. Then when we look at the old clade Protostomia, we don't recognize that as a valid clade. I'll talk about how that's been dealt with on the next slide. But the chordates, the echinoderms, hemichordata, and a couple of other phyla, very small phyla, belong to the clade Deuterostomia because they follow the deuterostome developmental pathway. So let's put in here other phyla, echinoderms, echinodermata, hemichordata, and a couple of very small clades that also belong amongst the deuterostomia. So what happened to the protostomia or the protostomes? Well, it suggests the DNA analysis, basically, gene sequencing analysis suggests that the protostomia don't represent a single clade, but represent two clades, the ecdysozoans and the lophotrochozoans. So these are derived the old protostomia. That's not very neat. Tough for you to read that, I think. Excuse me. So rather than having a clade protostomia now, no longer valid, we recognize the ecdysozoans and the lophotrochozoans. So the ecdysozoans are a group of phyla that share the common feature of having an external exoskeleton that they shed periodically through a process called ecdysis. So the shedding of the exoskeleton is required for growth. And it's actually triggered by a hormone. And the name of it escapes me right now. I used to know what it was because a colleague that I worked with over at UCF when I was on the faculty over there was working with fruit flies, and we sat and talked for a while about fruit fly development. And he went through the developmental pathway, and I learned the name of the hormone at that point, but I've forgotten what it is. Years since I've even thought about it. But there is a hormone that is found in arthropods, and particularly in insects, that is released and triggers ecdysis. That's the fancy name, the scientific name for the shedding of the exoskeleton. So you should be familiar with this. Here you can see, in this case, it looks like a cicada shedding its exoskeleton and emerging as the full adult form. It will take a few hours for it to stretch out its wings and for it to harden up the exoskeleton, which makes it incredibly vulnerable during that time period. So all arthropods that undergo ecdysis, which is all of them, always have this window of extreme vulnerability when they have just shed their old exoskeleton and they're waiting for their new exoskeleton to harden. When we talk about arthropods in chapter 33, I'll talk a little bit more about the consequences of that. But this is ecdysis. It's the defining feature of those organisms that we refer to as ecdysozoans now. The other group are called the lophotrochozoans. And there are two basic groups that make up the lophotrochozoans, and they are united on the basis of DNA sequencing. So gene sequences unite these together, even though they don't share common features that define them. So the Lophotrochozoans consist of two basic groups of clades. So the Lophophore clades are the ectoprocs and lamp shells. And again, when we talk about these in chapter 33, I'll go into a little bit more detail. So the ectoprocs and lamp shells share this common feature, this feeding structure called the Lophophore, which basically has a whole lot of these tentacle-like arrangements with lots of cilia on them that wave around and set up water currents that draw food into the mouth of the animal, which is at the base of the Lophophore. So this is for feeding. All the other clades share this trochophore larval stage. It's pretty much uniform. Now you can see that this is a larval stage that would tend to be aquatic. So those members of the Lophotrochozoans that are terrestrial tend to have either association with water or a grossly reduced larval stage because of the requirement to be in a marine or freshwater environment for the larval stage to be able to move and to feed itself. So we'll talk a little bit more about that when we cover these again, when we do chapter 33 in the next presentation. So that gives you an introduction to the animal kingdom and basically lays out the direction that we're going to take over the course of the next two chapters as we work our way through first the invertebrate side of the animal kingdom, which is by far and away the largest part of the animal kingdom, constitutes 95% or more of the animal kingdom as we know it today. So invertebrates are a huge part of the animal kingdom. And then in chapter 34, we'll focus primarily on the vertebrates, although we'll talk very briefly about the two invertebrate subphyla within phylum Chordata, which is where we reside as vertebrates, before we go on and focus on the vertebrates. So we still have a little bit more invertebrate coverage in chapter 34, but that's because two of the three subphyla within phylum Chordata are in fact invertebrates. So that just simply reinforces the fact that while we pay most attention to the vertebrates, in fact, the invertebrate side of the animal kingdom far outweighs the vertebrate part of the animal kingdom. So what are the likely directions for animal phylogenetics in the future? Well, there are two areas of focus. Number one is on where do the ctenophorans actually lie? So right now, the ctenophorans are considered to be more basal than the cnidarians. It's gone backwards and forwards on the nature of this relationship, in fact. Whether the ctenophorans are more closely related to the rest of the animal, to the bilateria, or whether it's the cnidarians. It's not clear. We don't have a lot of information that tells us what the nature of this relationship is. So there have been a couple of studies produced that have suggested that maybe the ctenophorans should be based back here as more basal even than the sponges. The problem with that idea is then that we would be placing a clade that contains true tissues more basal than a clade that has no tissues, because the porifera of the sponges are partially characterized by the lack of true tissues. And that creates an issue for this checkpoint or this branch point that represents the origin of true tissues. If we put the ctenophorans back here, then we're going to have the origin of true tissues occurring at least twice. And that then means that we have to rethink the whole nature of the animal kingdom. But again, this is based on only a single study. So the chances are really good that when further, particularly DNA sequence data is collected, the ctenophorans will no longer be, or there will be no indication that the ctenophorans are in fact more basal than the sponges, and will be restored back to this position amongst the eumetozoa, animals containing true tissues. The other issue is with these guys, the acellar worms, that are considered to be basal to the bilateria clade. Are they in fact bilateria, or are they basal to the bilateria? And actually lie more basal to the bilateria and represent a sister clade to the bilateria rather than being bilateria themselves. Again, this rethinking of the position of the acellar is a consequence of a limited number of studies looking at a limited number of genes and doing genetic comparisons. The chances are really good that once the access to more sequencing data, because of our ability to sequence genes more easily, becomes available, that we will again consider the acellar worms probably to be bilaterals. They certainly share a lot of traits with bilaterals in terms of development and body plan. So it is possible that these will remain within the bilateria, but a sister clade to the three main clades of bilateria, or a basal clade to the three main clades of bilateria, the deuterostomia, the lophotrochozoans, and the ecdysozoans. More evidence is needed, more sequencing data is needed. So that's the potential direction in which the studies will likely move, because these are questions that are being asked right now to try and understand better the phylogenetics of the animal kingdom. So one more concept check, and then we'll be done with this chapter. So what evidence suggests that the cnidarians share a more recent common ancestor with other animals than with sponges? So here we have to take a look at what we mean by eumetozoan versus metazoan. So when we think in terms of the sponges as metazoans, they possess no tissues. Cnidarians as eumetozoans do possess tissue. So that's one evidence that they are more closely related to the rest of the animals. A second feature is that cnidarians do have a body symmetry. It may be radial rather than bilateral, but they do have a body symmetry. Sponges do not. Now, that may seem a contradiction when you look at sponges, because some sponges do look like they have a body symmetry, but that's just simply the nature of that particular sponge. As a group, sponges have a random body plan. They do not have a formal body symmetry. And if we were on campus for lab, I could show you some body plans of sponges that are very random. They show no structured body plan. So that's again, another reason why cnidarians having a body symmetry are more closely related to the rest of the animal kingdom than to sponges. So that's it for chapter 32, a relatively short presentation compared to some of the others that we've had, and certainly much shorter than the 2 remaining presentations. Unfortunately, there's no real way to keep those short because these are very long, very information-dense chapters. So we'll just have to deal with them as they come. Anyway, as always, if you have any comments, questions, or concerns about anything that I've covered in this chapter, in this presentation, by all means, come and see me during office hours, shoot me an email, or in some fashion, contact me to get clarification on whatever it is that you're concerned about. Otherwise, I'll see you back here again for the next presentation when we will be looking at invertebrates. See you then.